my name is Yong Jian. I'm a PhD student from Department of Electrical and Computer Engineering, and I'm from the lab of general science of system biology. So here I want to introduce something about biology. So we want to put another puzzle to this workshop, which is biology. So although I didn't list the history here, but basically we are using the single-cell RNA-seq data to applying the well-developed machine learning models to data mining the interesting or active patterns that biology is interested about. And last 5 years since the company Untitled Genomics has commercialized the single-cell measurement technology, numbers of single-cell related research has been really enjoying a boom. And here I want to introduce some history about the development of the scTenifoldXct, which is the method that I want to introduce today for the cell and the cell communication a little bit. So we have the in our lab we have the scTenifold suite, and the scTenifoldNet is the cornerstone of this suite, and it has been developed by my senior and published in patents 2 years ago. The basic idea of this is to extract the gene regulatory network, the network between a pair of genes from the single cell data. So the single cell data is nothing mystery, but table. It's a table. It's a tabular data. It's a cell by gene data, or gene by cell, whatever you want to look at from different angle. And here we want to input two different single matrix to our scTenifoldNet method, and we want to compare our built gene here, our built gene regulatory network from there. So we had developed a couple of methods to derive the gene regulatory network from the gene expression. I'm not going into the details, but the main idea here is to we use the PC regression, principal component regression, to overcome the great sparsity of the matrix and also increase the speed of inference. So from there, we ask ourselves whether we can continue using this well-developed pipeline and to data mining. Another great attractive question for biologists is that whether we can learn the difference between the wild type of a gene, of a wild type of a condition, of one condition, and against the other knockout condition. So knockout in genetics is that you're gonna delete one or multiple genes from the animals or human beings that we want. And then we compare the functionality of these knockout samples to the healthy samples and to learn the function of genes. So here, the naive or the most intuitive way to achieve that is we just remove the links of the genes in the regulatory network that we construct and compare the difference between these two graphs. So this is another work of scTenifoldXct, and if you're interested, we published early this year, so you can check online. So this work is one of my recent work. It's scTenifoldXct. So here I listed some key features I'm gonna review after I introduce the workflow. So some background about the cell and cell interaction. So most of you are not from the biology. So here I listed some basic knowledge about this. So ligand and receptor are just genes. They're a pair of genes that people from the biology to learn that they are the sender and the receptor between two different groups of cells, and and when they talk to each other, the cell communication happens. And the baseline here, the baseline in this field before we started the research is that people just naively use the product of the gene expression of ligand and corresponding receptor as their score. If you treat the expression of ligand and receptor as a vector across cells, then this score would be somehow you can treat it like a dot product. And this is really a simple way to measure it because it just measures the similarity between the ligand and receptor. And in biology, we know that not only the gene expression, the policy of the ligand and receptor can represent the communication, but also their upstream and downstream genes in the network. So the motivation to develop the pipeline is that we want to use our already on-the-shelf PC regression method to get the gene gene network. And I also want to put some non-linearity to the score of the ligand receptor score. So in order to put the information of ligand receptor into the downstream manifold learning, we need to, if you're familiar with that, you need the correspondence between those features. So if you are not studying the multimodal data, if you are studying the two data sets from the same feature, your correspondence matrix would look like a diagonal matrix, indicating the similarity between your features. But here we use the ligand receptor product as the score in the correspondence. And instead of just using the mean, we put some variability. So it's like another version of the second moment to the correspondence score. So here, in order to learn the low dimension representation of genes, we denote H here. H is the stack matrix of HA and HB. A and B are the two conditions. The loss function is like this. So if the loss function consists of three terms, and the first two terms is to maintain the global similarity in the high dimensional data, and the third term is to try to maintain the correspondence of the two multimodal data. So the traditional way to solve this problem, if you're familiar with manifold alignment, is to use the eigen decomposition to decompose the design matrix with this WA and WB sitting in the diagonal and the correspondence S sitting in the off diagonal. We want to get the eigenvectors of that matrix. That's more time consuming, and if you apply the algorithm of eigen decomposition, you need your design matrix be symmetric, which will impose more restrictions to the computation work. So one of novelty here, I will try to convince you that instead we apply the neural network here. We just use the neural network, two neural network, HA and HB, and theta is their parameters to solve this problem, to solve this manifold learning problem. So after we solve that, we project our features from two conditions into a joint embedding. And then we somehow can calculate so here we select the Euclidean distance as the matrix test. So here, our test matrix Dij, gene I from condition A and the gene J from condition B. The distance of them looks like this. So intuitively, first time I saw this, you will say we can design a chi-square test or something like that. But it's not the same because we want to get the left tail of this distribution, while the chi-square use the right tail. So here we design another non-parametric test, and if you want to know the detail, you can look at the preprint. Probably we're gonna publish later. So this is the brief introduction of the workflow, and here is some case study using the real single-cell RNA-seq data. So okay, so it's a fibroblast that talk to the dendritic cells, and these two things are the ligand receptor. This paper they introduced in their research, so we validate the result of their findings, and we also compare our method to other benchmarks, and we see a lot of overlaps and some distinct findings, and also we evaluate using the ROC curve, and since the data is also unbalanced, so we also did the precision and the recall curves. And most of them, our method ranked the highest. Last one is about scalability. We put a different dimension of the data into our method, and we measure the time that they consume. And you see all of them are within the reasonable time. So lastly, I want to review some key features. It's a semi-supervised method, and we consider the underlying single-cell gene regulatory network to conduct the manifold learning, and this is and we use a neural network to solve this manifold learning problem. So that's all.
Thank you. Time for a quick question.
It's like you mentioned newly defined interaction score. It means the
it doesn't mean we integrate variability into the like this one, the second one.
Okay.